we'll make sure to share those at the end of the talk. All right, Roan, we ready?
I think just about. It's loading now.
Okay.
I see Dr. Riley is here, so she can grill Becca on some of the chemical principles.
Oh, I already had enough of that during my comps.
Yeah, she's already been through comps, so.
That's right. There's a limiting principle that takes effect, right?
Exactly.
Okay.
All right.
Roan, let's do it.
We're live. All right. Thank you, everyone, for joining us today on this lecture series, as we've had them for heading on a year now. We have two guests today, incredible alumni. This is the first time I think that we've had two people, Dr. Bob Brosh, 85, and Becca Lee, class of 2020. Becca has joined Dr. Bob Brosh doing medical research. They're involved with the NIA, and she took a fellowship with him. So part of wonderful connections that we've had through alumni. And I think he's trying to convince her to stay maybe a little longer with him as well. And Becca is going to be heading on to medical school eventually, whenever maybe he lets her go. So I'll let them tell you guys a little bit more about their stories and what all they've been working on. They have some incredible accomplishments, especially studying COVID right now. So if you guys have any questions throughout, please feel free to put them in the chat. And then at the end, we'll have some time to open up for some questions as well. But with that being said, I'll let Becca and Dr. Brosh go ahead and take over.
Okay.
Thanks very much, Rowan.
Thank you, Rowan. I'm going to share my screen. Let me make sure. Okay. Okay, can you all see my screen?
No, we don't see it yet. Oh, there we go. Okay. So now I'm going to go to view options, request remote control, request.
All right.
I should be able to, there we go. I'm going to grab the laser pointer. Okay. And yep. Okay, let me go back to the first slide. Okay, I think we're ready to start. Thank you all so much for coming to hear about our research on COVID-19. Really pleased, I know I speak for Rebecca Lee as well, that we very much appreciate Bethany College sponsoring this COVID-19 lecture series that started last fall. And, during the pandemic, it's been so valuable to hear different perspectives in so many areas pertinent to the pandemic. And special thanks to Roan Thrash, the alumni director for Bethany College, and also Joe Lane, provost at Bethany, for this opportunity. So the title of our talk is COVID-19 Life Cycle and Molecular Studies of the Novel Coronavirus. So this is our research team. We are in the Translational Gerontology branch at the National Institute on Aging, one of the 27 institutes of NIH. And my group consists of staff, specialists in biochemistry and genetics, and trainees, including Becca, who's doing a Postbaccalaureate Fellowship for 1 year in my lab, and we also have postdocs as well. So just to give you a little bit of background about the National Institute on Aging, our focus today in the talk is COVID-19, and that is relevant to aging. But sort of in a bigger picture, we're very interested in how genetics, environment, and lifestyle choices influence the aging process and age-related disease. So I would say that that's what drives all the researchers, several hundred at the National Institute on Aging, for what we do. But today we're going to specialize and focus in on COVID-19. You know, we see here the image of the coronavirus particle that has been such a culprit over this past year. And it is interesting that this week is the 1-year anniversary for the widespread lockdown in the United States. So it has been a long, unwinding road getting through the pandemic. So many issues have come up in terms of biomedical advances in understanding the coronavirus, societal issues, political issues, and this journey continues. And so having this COVID-19 lecture series is really valuable because dialogue is key to progress. Today, we'll be touching on a few different topics. I'll start by discussing a bit about the pandemic and some of the hot button topics that are coming up with vaccines and coronavirus variants. That will lead into a discussion about the coronavirus genome. The coronavirus is also known as SARS-CoV-2. And from there, Becca will discuss the coronavirus life cycle. She will focus in on one of the critical factors required for the replication of the coronavirus. It's a helicase protein, and we'll describe that, known as NSP13. And then to sort of wrap up, she's going to give a broader view of her experience as an NIH Postbaccalaureate Fellow. So I guess we'll first consider the cornerstones to confront a pandemic. And those have really come out in the past year, thinking about the community and behavior-based actions, the social distancing that Anthony Fauci and the staff have requested, to wear masks, hand washing, to do those things to prevent the spread of the virus. A second layer are the antiviral medicines, and these particularly hit a high profile early on in the pandemic with the discovery of Remdesivir, a drug that may be important to block coronavirus replication in individuals that have been infected. There's also the interest in monoclonal antibodies, and these would be to reduce morbidity and mortality of infected patients. And then most recently, with the push from pharmaceutical companies on the development of vaccines from Moderna, Pfizer, and most recently Johnson & Johnson, this has created a lot of excitement, and it's starting to happen. And these vaccines would reduce transmission and ultimately eradicate or come close to eradicating the virus from the population. So let's just talk a little bit about COVID-19 vaccines and the challenges there, because it relates to our research efforts that we'll talk about later on antiviral medicines as a supplement to the vaccines. We know that the production and distribution has been an astronomical challenge, but thank goodness it's starting to move, and more of our friends and neighbors, family members are getting vaccinated, which is fantastic. The question comes up, how long will the vaccine protection last? And if there's going to be a requirement for booster shots down the road, and also coronavirus variants, if those vaccines will have efficacy. We'll talk about that. Who will be protected? So we know that there are special populations. Age is a major risk factor for the severity of COVID.
Actually, Bill Conley said he sent the invite 2 weeks ago to your Cabell email.
I think we need to mute.
It's in the medical affairs conference room. I said, well, what if he's still here seeing patients and he can't report to that room? He said, then you could call your cell phone. You could call his cell phone, but that you should definitely be involved if you have an opinion about it.
Paul's iPhone
Sure. I
mute.
think you probably will.
Okay. Okay, there we go. Thank you. And comorbidity, individuals who have other diseases, including in those who are frail, as well as special populations like pregnant women and how well the vaccine will work there. So the other aspect is virus transmission and how successful that will be to achieve that highly desired herd immunity that we've heard about even in popular news so much. And finally, for challenge to vaccines against COVID-19, these emerging coronavirus variants that may be partially resistant to the vaccine and the requirement for development of further vaccines. So thinking about the vaccines, and now we're going to go into a little bit more of a molecular coronavirus perspective. We know that coronavirus has a particular appearance shown on the left side of the slide with those spike proteins that form that sort of crown appearance of the coronavirus. And the engagement of that spike protein with a receptor on the cell membrane of the human host cell, often a lung cell, because they're enriched for these ACE2 receptors that specifically bind the spike protein of the coronavirus particle. And as it turns out, there can be monomers. It's actually the trimeric form of the spike protein. 3 of those monomers working together that it forms the high affinity interaction with the human cell membrane receptor. This is sort of important from the vaccine approach, and we'll talk about that, but also the variants. So one of the mutations that has been identified in this coronavirus spike protein is centrally positioned in the tetramer, or in the trimer, and it's called D614G, and aspartic acid is replaced with a neutral glycine. So aspartic is negatively charged. And what this ultimately does is it changes the conformation and the flexibility of the spike protein, and that increases the affinity to the receptor on the human cell membrane, so increasing the probability of infection. And this becomes important from an epidemiological standpoint when you look at the global spread of the coronavirus going back to 2020. We remember that in late February, the outbreak began and the virus, those infected just went up exponentially. And that corresponded with one of the variants at this particular residue that I was describing. It's this glycine variant at 614. So, by late June, almost everyone who was infected with this particular coronavirus variant. And even in more recent studies, and there have been a lot of labs that have tried to understand this, and they're making nice progress. So this just came out a week or so ago, looking at in model mammalian organisms, including hamsters, that were exposed to the coronavirus inoculum, either the G variant or the D variant. And those inoculated organisms were housed in cages with uninfected hamsters. And then what was done were nasal swabs were taken every 2 days for out to a couple weeks. So initially it was a 1-to-1 inoculum of the 2 variants. And what you can see is that orange, the 614G variant really, really persisted in the infected ones. And importantly, in those contact animals that were infected, it was the G variant that was persistent. So this is telling us that there's an enhanced transmission property of one of these particular variants. And this is really important information that may be informative moving forward on the newly described variants. And some of those SARS-CoV-2 variants have been described, and they're from different countries of origin, the United Kingdom, South Africa, Brazil. And there is still to this day a lot of studies on the behavior of these variants in terms of their transmissibility, their contagious degree, their virulence, how severe the clinical features are for those people that are infected, and also vaccines, how successful they will be in neutralizing these different coronavirus variants. So this is, the jury is out, I would say, but it's a very important area of study for both clinicians and biomedical researchers and virologists. So a question, I'm trying to cover some of the bigger view here, and then we'll be going into a little bit more of the life cycle and molecular functions that Rebecca will cover. So why do some infected people get very serious COVID-19 disease symptoms, whereas others are only affected more mildly? And we know that there are two major classes. There are patient risk factors, and then there are genetic risk factors. So patient risk factors, for one, is definitely age. And that's why we're particularly interested in this disease from the perspective of the National Institute on Aging, that there's an increased susceptibility with age to infection and also to the clinical features, the severity of disease. We also know race, ethnicity, vitamin D levels, cigarette smoking, and diet, to the extent of obese individuals are more prone to the severity of the symptoms. And then lastly, we can think about comorbidity for a patient risk factor, other organ systems that are already compromised in the individual: diabetes, hypertension, cardiovascular disease, chronic lung disease, or chronic renal disease. And then switching gears, genetic risk factors. Recently, there's been nice progress in understanding blood group locus susceptibility. And on the lung cell, which is one of the major cells that is infected because it has a receptor for that spike protein of the coronavirus particle, it turns out that the A antigen that is inserted into the plasma membrane, the cell membrane of human lung cells, shares a lot of homology with the normal receptor for the spike protein. So by it being on the surface, it's just increasing viral load, the probability for infection. There's also the immune response, depending on that acute response, known as the innate, and then a later, more specialized response to the coronavirus antigens that are introduced into an infected individual, that adaptive response. And lastly, an imbalance in the immune reaction, if there's an excessive inflammatory response, and this is known as the cytokine storm, that there may be actually an attack on normal human cells, and that's sort of misdirected. So I had mentioned about age as a risk factor. We know that for younger individuals, they typically get, they're asymptomatic, or they only get mild forms of the disease, whereas individuals over the age of 60 have a more severe phenotypes, and those are shown here. And then with advanced age, you can have acute respiratory disorder syndrome, cardiac problems, other organs, including the brain, and then this can also lead to death. So there's definitely an age component. Age is a risk factor for the severity of disease. The SARS-CoV-2 genome of the novel coronavirus is one member of a family of RNA viruses, coronaviruses, that cause other sicknesses or disease, including the rhinovirus for common cold, HIV implicated in AIDS, the Zika virus, influenza that's responsible for the flu, Ebola, and then of course SARS-CoV-2 of causing COVID-19. And one of the impressive features is that the SARS-CoV-2 genome is significantly longer than the other coronavirus genomes. And down here, you can see that the coding sequence that actually makes coronavirus proteins in that genome, it's very, very dense. There are lots of genes being transcribed and translated to make proteins, including those that are involved in replication in the first half, and those that make up the protein coat of the virus, the sheath, as well as accessory factors in the second half. Interestingly, because the coronavirus has a proofreading subunit, it incurs relatively few mutations, but it does have some, and we know those can have quite strong implications, as I discussed earlier. So just a little bit of the replication mechanism for the novel coronavirus. It's a single-stranded RNA virus. When it infects the cells, it gets converted into duplex RNA, double-stranded RNA, and this can either be transcribed to make so that the host cell makes proteins by translation. These viral proteins are critical to the life cycle of the virus, or the molecule gets duplicated so that you have more double-stranded and be able to replicate the virus. So this is the essential functions of the viral RNA in its single-stranded or double-stranded states. And this is this slide illustrates that compared to conventional double helical DNA shown on the right side, where it forms this nice 2 strands in a helical nature, 10 base pairs per turn, RNA has a more compact or squat architecture where it's actually 11 base pairs per turn. And that may not seem like a lot, but you can see from these 3-dimensional structures that it's actually quite dramatic. So the coronavirus nucleic acid in its double-stranded form is dramatically different from even the human genome DNA in terms of structure. And beyond that, even the single-stranded RNA of the coronavirus can form these really bizarre, complex structures. We call these hairpins or cruciform structures that are really quite complex. And these may have different roles in protecting the coronavirus from nucleases, because the virus, from its selfish standpoint, wants to keep its genome intact. It may be important in the stability or the translation of this RNA to make coronavirus proteins, and it may even play a role in immune evasion. One of the areas that we're interested in is this idea that these regions might be susceptible to mistakes, mutations, and we know that those are very important for these coronavirus variants that we talked about. There must be a mechanism to resolve these RNA structures so that the virus can be replicated. And that brings us to a protein, an enzyme that my lab is studying. It's a molecular motor protein, a helicase, that disrupts these many hydrogen bonds between complementary bases. And so you need an enzyme to do this, and that is a helicase. And as it turns out, the coronavirus genome encodes a helicase of its own that is absolutely essential for the replication of the coronavirus. And that helicase is called NSP13. So lucky number 13. This is a critical player encoded by the coronavirus required for the replication of the virus. So these helicases that we've been studying for over 20 years, there are helicases encoded by the human genome. They're actually very ubiquitous in every organism known, unicellular or multicellular. And these helicases have been studied. They can unwind duplex DNA or RNA, depending on the helicase, by different mechanisms. One is a simple inchworm mechanism where there's a head and a tail, and in a manner dependent on ATP hydrolysis, the energy, it simply moves through the duplex It translocates forward and disrupts those hydrogen bonds. You can have functional cooperativity among helicase molecules, sort of analogous to train locomotives pushing up a hill, pushing each other, and that makes a more processive helicase. There's a rolling model in which the helicase entity is a dimer, and actually the subunits roll over each other. And the advantage to this is that you always keep the helicase tethered to the nucleic acid that it's unwinding. And lastly, there are hexameric ring-like helicase structures that form a donut. One strand goes through the middle and the other strand is extruded out. So this is very interesting as we study the NSP13 helicase that Beck is going to be telling you about, because understanding its mechanism of action is vital to understanding its role in coronavirus replication, and also raises the possibility that this NSP13 helicase may be a potential target for the development of antiviral medicines.
All right. Thank you, Dr. Bob. Just a few points that I'm going to key in on during my presentation is going over the SARS-CoV-2 life cycle. And from here, I will go more into the specific research that I have been conducting in the lab to characterize this NSP13 helicase. Once I get through that, I'm just going to tell you guys a little bit about my experience that I've had thus far as a postbaccalaureate fellow in Dr. Brosh's lab. As I'm sure you guys are all aware, there's multiple viruses available to the public right now. However, active research is still needed to understand the coronavirus life cycle and its replication complex. And once we have this, this could potentially lead us into discovering various drug targets for therapeutic strategies. In this slide, it's just depicting an overall view of the life cycle of SARS-CoV-2. It's suspected that the virus initially infected bats and has mutated through intermediate hosts to be transmissible between humans. Contaminations in the environment are typically found in respiratory droplets, and once those respiratory droplets have entered into the human airway, they travel down until the lungs. After they interact with a specific receptor on lung cells, cellular entry can occur. After the engulfment of this virus, there's a replication process and gene expression that needs to occur for new copies of this RNA virus to continue to infect. In addition to this, there's proteins that are needed for the structure of the particle, as well as just other accessories. With these proteins and the viral copies that have been replicated, there's a packaging process and prep for particle release. And once the particle and the new copies of this virus have been released from the cells, they are either able to insert into the body and infect more cells, or they are expelled back into the environment. Like Dr. Brosh had mentioned, one of the key components necessary for the SARS-CoV-2 survival is this RNA encased within this particle. There are multiple proteins that are encoded for this RNA, and a few of them I'm going to point out here. They're known as structural proteins, and they have to do with the integrity of the cell. And you have the N protein, the M protein, and the E protein. And one of the most crucial components of this particle is the spike, the S protein. And like Dr. Brosh mentioned, it is necessary for entry into lung cells. So as this particle finds and locates the ACE2 receptor, it binds to form a complex. And as this occurs, a protease enzyme attaches itself to this spike protein and looks for a specific interaction that it's able to cleave. This cleavage allows for the spike protein to decapitate and expose some fusion machinery that's encased within it. And from here, this fusion machinery is just going to reach out and insert itself into the lung cell membrane. And as that happens, it's going to pinch together and morph into one large membrane that forms a channel to allow for these RNA viruses to enter into the cell. Once we're in the cell, a lot of different things occur. Since the RNA virus is a positive sense single-stranded genome, the first thing that it does is it translates into these viral proteins. And these viral proteins incorporate themselves into a replication process that's able to take that viral RNA, the coronavirus RNA, and replicate it into new copies. From here, still hijacking the host cells, different machinery, the RNA virus is prepped and packaged into vesicles. In addition to this, those viral copies are also used to produce some of the structural and accessory proteins needed for the survival of the virus. And after these incorporate into the same vesicle and start to form more proteins of the virus, they are able to exit the cell and continue to infect and spread. Now that we have a little bit more of an understanding of the overall life cycle of the SARS-CoV-2, one major component that I want to key in is this replication process. As you can see, there are a lot of different non-structural proteins that make up this complex, and each of them have their own special functions and responsibilities for the continuation of the virus. However, I'm just going to key in a couple. First off, this NSP12 is also known as the RNA-dependent RNA polymerase. And what this protein does is it takes this negative sense strand and it uses that as a template to make more of that positive sense RNA that is then able to form new viruses. In addition to this, another key protein is the NSP13 helicase, and that's the one that we are most focused on in Dr. Brosh's lab. You can appreciate the location of this helicase as it is kind of the lead of this complex. So as it unwinds this duplex and separates the hydrogen bonds between the complementary strands, it's able to directly feed this negative sense strand into that RNA polymerase to form new proteins. In addition to this, the NSP13 it acts as a molecular motor and has ATPase activity. In order to actually start running experiments, we actually have to have the protein purified. And this is a process that actually does take place within our lab. The first thing that happens is we grow up insect cells and we overexpress our target protein. And from there, we're able to use affinity chromatography to select for specific tags within that NSP13 plasmid to get a clear and clean protein. And one way that we're able to track this process, the purification process is lengthy. It takes about 2 full days of work. So after each fraction is collected from the column, it's loaded into a protein gel. And from here, a current voltage is run through the gel to allow separation between the fragments as it travels from negative to the positive terminal. And as you are able to finish the gel, you are able to see the purification and the purity of each step of the process. Josh Summers, the biologist in Dr. Brosh's lab, has really mastered the art of this purification process. The data that's shown here in this gel is actually from the NSP13 purification. Each of these fractions are just, like I said, different steps of the process. We use specific resin beads in these columns to end up with this purified protein over here. And that is our NSP13. One way that we are able to test for the activity of this helicase is through helicase assays. And we are able to track this through a radioactive labeled duplex. And so in this case, the star is representative of where the radiation material is on this duplex. This substrate is incubated with this helicase for, depending on the reaction conditions. In this case, it was incubated at 15 minutes for 37 degrees. After this, the reaction is quenched and it's loaded through a protein or gel. Similar to the protein gel in the purification, a current is ran through and the separation between the components of the samples is looked at. After that, we're able to image it and look at the activity of the helicase as well as quantitate what it's showing. In this instance, this is just a simplistic image of what a helicase assay may look like. And so up here we have your intact duplex link or duplex substrate, and that means and that is typically not unwound right away. In addition to this, in the assay, we're scanning and looking at this single-stranded labeled product. And as you can see, as we increase the time here, more of that product is evident in this gel. Although the NSP13 helicase is an RNA helicase, it still has the ability to unwind DNA substrates. The data that's shown in this gel is with a 19 base pair DNA substrate. The data that's shown with this gel in this graph is the NSP13 as a function of protein concentration. So in each of these lanes, we are increasing the amount of protein that's present in the solution. And what we're seeing here is as we increase this amount of protein, we are able to see more of the final product, more of that unwound substrate. And by quantitating this data, we're able to present it in a graph. And what you're seeing again is as you increase this NSP13 concentration, you're also increasing the amount of DNA substrate that has been unwound. In this particular instance, there is about 0.5 nanomolar concentration of substrate present. So when there's equivalent concentration of that NSP13 enzyme, we see about 50% unwinding. And as we increase the NSP13 present, we see a substrate depletion effect here. In addition to this characterization of the NSP13, I also was able to look at its helicase, its dependence on ATP hydrolysis. And each of the different lanes here are just looking at different energy sources, different nucleotides to see whether it is dependent upon ATP or not. And what we did find was that it was, and that's evident within this ATP gamma S lane. In addition to this, the majority of the energy sources that were used were optimal. However, this DTTP was just a little bit suboptimal. Switching gears a little bit, now I'm going to talk about the NSP13 as it unwinds RNA substrates. And these are the 4 specific substrates that I have been working with thus far. They have a 26 length nucleotide tail, and then they have an increased duplex length starting at 13 base pairs and increasing by 3 all the way up until 22. And in this instance, the radio label is now on this blunt 5' end of the duplex instead of on this tail. The data that's shown in here is just, again, the NSP13 activity is a function of protein concentration. This specific gel is looking at a 16 base pair duplex. And we were able to actually achieve higher protein concentrations in this instance. And what you're seeing again is just as you increase the amount of enzyme present, we see more of that product, that single strand, meaning that the helicase was able to unwind this duplex. In the graph over here, it's just showing the 4 different substrates of the RNA duplex. At these lower lengths, there is a pretty consistent unwinding. There's not a significant difference between the 13 base pair length to the 16. However, as we increase to that 19 base pair length and additionally to that 22 pair, we see a drastic drop in the ability for that NSP13 to unwind the duplexes. Continuing to look at these specific substrates, I then looked at the NSP13 activity as a function of time. And I was able to do this through a single turnover kinetics analysis. And basically what the data is showing here is that the NSP13 is able to unwind in these specific conditions a lot more effectively than these larger duplex lengths, the 19, the 16, and the 22 base pair. And what this data is showing is that the NSP13 is not a processive helicase. And a processive helicase means that a helicase is able to attach to a duplex and it's able to unwind the duplex in its entirety. However, the NSP13 is not processive, meaning that the enzyme does not stay attached to able to unwind the duplex. With this and with knowing that the NSP13 is not as processive as it could be, it suggests that there are factors within the human cells that help to stimulate the ability for it to unwind the long RNA genome that is encoded by the coronavirus. In Dr. Brosh's lab, we're taking a 2-armed approach to this to analyze and look for stimulants. One way that we are doing it is we're taking human cells and injecting NSP13 affinity beads in here. And what these beads are doing is trying to have an interaction between cytosolic factors that can be found in the human cells. From here, the cells are going to be lysed, and then we hunt for specific interactions between these, the helicase and potential host proteins. In addition to that, we are looking at known RNA binding proteins to see if they also increase the ability for this NSP13 to unwind. One that we're very interested in is the NSP9, and that's the RNA binding protein that's encoded by the coronavirus. In addition to this, we will be testing other known human RNA proteins that are found in cells. So kind of taking a step back and understanding how all this data plays into our research in the coronavirus, we are now looking at potential antiviral medications, and just researchers in general are looking for medications that are able to cripple the virus to help with the ongoing coronavirus pandemic as well as future ones. There are a couple of strategies that researchers are using to look for these drug targets. With the vaccines, they're looking at preventing the virus from entering the cell. However, if the virus is able to enter the cell, some researchers are looking at shutting down the virus by targeting these host proteins that are vital for the replication of the genome. Another way that researchers are looking at drug targets is through reducing the immune response. So those individuals that have severe symptoms, is there a drug that can come in and alleviate it? In addition to that, another one is looking at defective viral replication. And that's the one that we're most focused in on in Dr. Brosh's lab currently. There has been some initial research to find a medicinal compound, Remdesivir, that has been seen to have antiviral properties. And this specific compound is a nucleotide analog. And so what it does is if it's present with the coronavirus, it acts as an inhibitor for that NSP12, that polymerase for the virus. And in turn, it is needed to terminate this RNA synthesis. Some clinical research has been done with remdesivir in patients with severe cases of COVID-19. This data was released in 2020. However, continuing research with the remdesivir in the clinical aspect is needed. However, in this case, there was seen to be a decrease in the mortality rate as well as the duration of the illness when present with remdesivir. One aspect that researchers are really keen into nowadays is looking at small molecules as potential drug targets for these antiviral medications. And that's what we've started to work on in Dr. Brosh's lab. So we have a drug library in-house right now that has about 1,500 different compounds. And what we're able to do with these compounds is run them through the Helicase assay with NSP13 to see if there is inhibition. So if there is inhibition, that could lead to the virus's replication, propagation, infected to be crippled. A key theme that's very prevalent nowadays within the clinical field is drug synergism. And what this is when there's an interaction between two or more drugs at the same time, and to see if their effect is greater than the sum of the individual drugs individually. It's suspected that the helicase and the RNA-dependent RNA polymerase within that replication complex do come in contact with each other. And so by introducing a helicase inhibitor as well as remdesivir, which is actually the inhibitor for this NSP12 molecule, what we're hoping to see is this inhibition of replication. So even though there has been vaccines created, active research is still required and necessary to fully understand the coronavirus itself and to prepare us for future ones as well by understanding the life cycle of the virus as well as understanding the viral replications. This could lead to opportunity for us to develop antiviral medications. In our lab, the one we're most specifically targeting with these small molecule inhibitors is this NSP12 or NSP13. And hopefully with our contribution, antiviral medication will be widely available to the public over the counter to help reduce the symptoms of individuals that do have this coronavirus. Right. Okay, so switching gears a little bit, I'm just going to talk to you a little bit about my experience here at the NIA working as a postbaccalaureate fellow in Dr. Brosh's lab. So our lab, the NIH, is on the Johns Hopkins Bayview campus in Baltimore, Maryland. This is just a picture of our lab. This is what a typical day looks like for me in the lab, where I do work with radiation. I have to be in a specific radiation room. And to limit the exposure to the radiation that I'm working with, I work behind this plexiglass box. In the instance, the radiation that I'm working with, it does stop that penetration. So I was able to start my work here in the lab in October. So I've only been a member of Dr. Brosh's lab for about 5 or 6 months. I've learned a lot during my time here. Not only have I learned to be a full adult and live on my own, I also have learned how to conduct proper laboratory research and learn skills that are needed for the techniques that I've been using. In addition to this laboratory work, I've also been able to have access to a wide variety of literature to be able to study the molecular biology and biochemistry of the SARS-CoV-2. Although I am pretty independent in the lab and the experiments that I am running are with me, there is a huge team component to this research, and it wouldn't be able to get done without this team aspect. Outside of the lab, I've had a lot of opportunities to network with other postbaccalaureate students as well as postdocs and kind of get a feel for what they're doing in their journey. Outside of that, I have had the opportunity to talk to varying staff and aging experts. In addition to this, I've had the opportunity to participate in some community outreach. This past week, I was able to give a talk to student or school-aged children about the coronavirus. And just going on from here, I continue to complete work with Dr. Brosh's lab for the time being. After my time here at the NIH, I have been accepted into Marshall University's medical school, and that's the path that I want to take. However, I really plan to incorporate the skills that I have been fortunate enough to learn here at the NIH into my future as a physician.
Thank you, Becca, for that presentation. It's really very exciting. And I should point out that the data she showed is data that Becca generated in the lab. So this is great. I really appreciate that, Becca, and appreciate you having you in the lab. And it just goes to show the fantastic students that can come out from Bethany College with that STEM training. I should recognize that, as Becca said, it's a team effort. So we have a fantastic biologist, Josh Summers, who's really a master of this protein purification and enzyme characterization. Josh did his bachelor's at Northwestern University in biochemistry, and he's also earned his master's in Johns Hopkins in biotechnology. So he's a partner, a longstanding partnership between he and I on the study of helicases implicated in diseases. Arindam Dada is a postdoctoral fellow who's doing some really promising work also in a premature aging disorder, and he's involved in the COVID-19 studies. So this is a team effort, and it's splendid to be working with such terrific colleagues. Lastly, I'd like to thank Bethany College for their terrific training that both Becca and I received during our times there. Special thanks to Joe Lane, the provost and dean of faculty, who is the coordinator of this COVID-19 lecture series. It's really an innovative and a creative way, and so valuable because we know that communication and learning are key to beating this coronavirus and getting out of this pandemic. Also, special thanks to Rowan Thrash, the Director of Alumni Engagement, for all his help in keeping us involved and reaching out to alumni for initiatives like this. I should also finally recognize the National Institute on Aging. This research that Becca showed you and is ongoing is supported by a special COVID-19 project grant. And I'm delighted to say that we received a new grant to continue this work, an inter-lab grant in which we'll be collaborating with some other key NIA personnel on this COVID-19. So we're really excited about the direction of the project that's supported by NIH and the Intramural Research Program. And with that, we'll be happy to take questions, both Becca and myself.
All right, thank you guys. We have a few questions in the chat already, and if anyone else has any, please feel free to, you're more than welcome to unmute yourself and ask a question or put the question down in the chat. So we have a few. Dr. Brosh, Martha has asked how your research was impacted with the onset of COVID-19.
That's a great question, Martha. Thanks, and hello. So, you know, at the Biomedical Research Center, Rebecca had shown a picture of the lab building. So we had to reduce occupancy, and that continues now. Fortunately, because we had a special grant on COVID-19, it allowed our laboratory group to have access to the research lab, which we needed, whereas other researchers at NIA were not able to. That was in the early phase of the pandemic in the first few months. As time has gone on, our scientific director and the chief executive officer of NIA have developed a really rigorous plan so that we minimize lab occupancy. For example, one person working at a lab bench, a limitation to the number of people that are actually in the building, and a lot of rules and regulations which are necessary to have a safe environment. I'm just thrilled that we continue to have our trainees working, and as Becca said, independently, but also supervised in a team approach, and be able to move this project forward. Thank you.
All right, and there's another question for Becca in here. There's a little blurb. This is like oral comprehensive exams all over again, Becca. So I don't know if you saw how homologous is the viral helicase with human helicases, and could this be an issue with potential side effects in humans?
Unfortunately, I am not the expert in this field. That I think that would have to be a question for Dr. Brosh. I'm so sorry.
Right. Well, I think the question, maybe to clarify the question, is, you know, with the helicase inhibitors that you're investigating, and Becca has started that project, the small molecule screen to identify potent and specific inhibitors. So it's very important that if we're going to go after NSP13 as the drug target to cripple the replication of the virus, that as the person asking, these inhibitors cannot cross-react and have an inhibitory effect on helicases encoded by the human genome that have normal functions in DNA repair, for example. And so we're very much dialed into that, and that will be part of the secondary screening. Once we identify hits in the primary screen, those compounds will be tested by for their effects on the unwinding reactions by a battery of helicases, human helicase proteins. And the other thing I would add here is that while we're starting with a focused NSP13 library of about 1,500 compounds, we've established a collaboration with an NCATS, a core facility that has the robotics that you saw in that picture that Becca showed. And that will enable us to look at hundreds of thousands of compounds. So we're really excited about this project, and we're definitely dialed into the specificity of the small molecules, as the person asking pointed out. Thank you.
Fantastic. Becca, I had a quick question myself. Just maybe I've been in comps too. I'm not going to come up with some crazy question that I probably couldn't even come up with anyways. Okay. But what advice would you pass along to, I know some of the seniors and juniors and so forth here, but what advice would you pass along? Because, being the chem major and in those pre-med tracks, like, what advice do you have for as far as fellowships, taking a gap year, med school? What was something maybe you would tell yourself now for when you were a student or so forth?
Well, initially, whenever I first went into Bethany College, my goal was to actually graduate within 3 years and immediately start med school that fourth. So at the time, whenever I was studying and going through this whole thing, I was still, I was just 20. I hadn't even had my 21st birthday yet. So I was still young and I still had a lot to learn. And as I grew, I decided, I think a gap year would be the best thing for me, just in terms of like taking a step back and enjoying myself before I go on to medical school. During that time, I was being a substitute teacher. I was working in a nursery in preschool with young kids while studying for the MCAT, and Dr. Brosh reached out to me over the summer, and I jumped right on that. I think a thing to remember is that you're still young. You can have time to have some time for yourself, and a gap year I would highly suggest. But also with that is being flexible with your plan. I didn't think that I would be doing what I'm doing today before med school, but I'm very fortunate that I am. And I feel like I'm able to grow individually, but also in an academic standpoint through this experience.
That's great. Well, I know I can speak from, our peers and friends, especially the ones that we have similar, that you represent us super well, and we're really happy that you found that place there with Dr. Brosh. So Dr. Lane, I don't know if you have anything. I don't think we have any more questions in the chat. We can absolutely pass more along. If anyone has any questions, please feel free to email Provost Dr. Lane or myself, and we can absolutely put you guys in contact, or pass those questions along as well.
Yeah.
But other than that, Dr. Brosh and Becca, thank you guys so much for the time, the preparation time, just willing to be with us and share everything that you guys have been working on and gone over. It's really, really incredible, and it's awesome to have two alum working together and providing these connections. And I'm excited to see, Becca, where you head, and we'll both maybe be Marshall grads as well. We stick with the green and white colors and the bison.
Yeah, definitely.
So go ahead, Ron.
No, that's okay. You can go ahead.
Well, I know that people on campus will be jumping off because 12:30 classes are gearing up, but if other people want to stick around and ask questions, I certainly do so. And I want to remind people that the talks are continuing, even though they're not all on COVID these days. Our next talk actually will be next week. We'll have a novelist on, Maricel Vera, reading from her work. She's a novelist based out of the Pittsburgh area and has spoken on campus before, but this time by Zoom. And you can join us for that, as well as another big one to look for later in the semester is Joan Maloof, the executive director of the Old Growth Forest Network. Will be talking for Earth Day on April 20th, which is another talk. We'll get those announcements out for people. We love having you join us, and we're glad you're here. And Becca and Bob, thank you so much. We're very grateful for your work and for sharing, and wish you all the best. We'll give a minute if people who are lingering want to ask one more question before they go. And thank you all. Have a great Tuesday afternoon. I hope it's spring where you are.
Thank you.
Thanks, Joe. Thanks, Brian.
Bob, I'm wondering about before the virus, what was your team studying, and how will that come back again, or what's your plan and what's the plan?
Right. So this is the COVID-19 is one project that we have going in the lab. We have a few projects moving simultaneously. So one of the major areas, and this is a great question, Martha, that we focus on is premature aging disorders. So these hereditary diseases that are characterized by accelerated aging. So we've seen things on the news, the popular news of progeria. As it turns out, the mutations that are inherited responsible for the disease are in helicases. They're in helicase genes encoding human helicases that have important functions in DNA repair, in replication, in transcriptional regulation. So that was a nice foray for us to focus on this particular class of enzymes. So we do the biochemistry, we do the genetics, not only with human cells, but also in model organisms like mice, to try to understand what are the functions of the normal helicase gene and what pathways have gone awry in individuals that have these premature aging disorders due to the helicase gene mutations. So we continue projects in that. And, you know, if I would have told you that I could build a career after Bethany College studying helicases the whole way, I would have never guessed it. And now I think I've got Becca, you know, I'm worried that she's going to steal my job in the future here because she's such a natural in the lab. They really are a fascinating class of enzymes. I could talk about it all day, but maybe Bethany's going to get overcharged in Zoom fees or something like that. Thanks so much for the question. I hope to hear from you all. If anything comes up, as Roan and Joe said, please don't hesitate to get in touch and they'll, you know, we can communicate.
Yeah, and Bob, we hope you and Becca are both back on, able to be back on campus doing stuff, live action with students here very, very soon. We look forward to that.
Would love to.
And Becca, you know, hey, best of luck in whatever you decide next year, or, you know, if med school's 2 years out, but if we can do anything to help you, let us know. Okay.
Thank you so much. My younger sister actually is going to be a freshman there next year. So we're continuing on the legacy of Bethanians in my family.
Fantastic. Yeah. Is she also somebody who sees herself going medical researching directions?
Yes, she is. She's
sure
very involved in STEM.
Dr. Riley and Dr. Brothers are ready to take her on. That's fantastic.
That's great. And we have a summer student internship program. Yeah. That we actually had a Bethany student a couple summers ago, Haley Thompson, who's now in veterinary school at Ohio State, and she did some fantastic work. So that's another trainee program that we have at NIA. That's a good opportunity to get hands-on experience, because that's what we really emphasize in these research internships.
Wonderful. Yeah, that's great. We will keep sending them, I promise. Hey, everybody have a wonderful, wonderful day. And thank you all so much. We were really glad to have this event. And the recording will be available on the website as well. So you can go back and watch it if you want to see it again, or share it with somebody who didn't catch it the first time. Hope you have a good one.
Okay, awesome. Thanks, guys.
Thanks, everyone.
Thank you, everybody.